Hello and welcome to another lecture from the UW Synthetic Biology lecture series. Today we will be discussing the article titled Robust Multicellular Computing Using Genetically Encoded NOR Gates and Chemical Wires. This article was written by Tamzir, Tabor, and Voigt from the University of California in San Francisco, and it was published in Nature Letter in January 2011. So in this article, the authors study the process by which they combine a simple genetic circuit with quorum sensing to produce more complex computations. Alright, so first they construct a simple NOR logic gate in an E. coli, and that is depicted right here. So the purple bordered thing is an E. coli, and they have two tandem promoters that function as input to drive the transcription of a repressor, and this is a repressor. And this repressor inactivates another promoter that eventually serves as the output. So if that's the promoter, so this repressor inactivates this promoter, and this is the output, which can be yellow fluorescence protein, say YFP. So this is the functioning of a basic NOR logic gate. Next, they arranged these E. coli colonies with these NOR gates that I just briefly explained in different spatial organizations. So say this is an E. coli colony with a NOR gate, so is this, and so is this. So the external inputs result in outputs which act as inputs for the other set of colonies, and the output from this red cell acts as the input for the blue cell, and the same here, the output from the purple cell acts as the input for the blue cell. And by spatially configuring all of these cells with these NOR logic gates, the authors were able to come up with several two-input gates, such as the XOR and equals gates. They were able to achieve strong and robust responses, and with like almost 500 to 300 times fold changes between the on and off states of these cells. Now this work is important because the authors were able to elucidate the design rules by which simple logic gates can be harnessed to produce diverse and complex calculations by rewiring the communications between these cells. So these resulted in different logics such as the AND, the OR, the NAND, the NOR, the XOR, the equals, just to name a few. So let's take a look at the results now, and we'll scroll down, and this is figure 1 from the article, and as you can see here, figure 1a is the truth table for a NOR logic gate right here. So the NOR gate is only on when both the inputs are off, and in any other situation the NOR gate is always off. So if both the inputs are on, the output is always off, and even if either input is on in a NOR gate, the output is also always off. So figure 1b is the genetic circuit of a NOR gate, which was something similar to what I depicted earlier. And so you have, just to explain again, you have two promoters in tandem, and when both these inputs are on, this will transcribe for a repressor, which will then act as the input for another promoter, uh, and it inactivates it, and that serves as your output. So figure 1C describes the um, stepwise construction of an OR gate, with PBAD and PTET as the input promoters and yellow fluorescent protein as your output gene. And now, PBAD is activated in the presence of arabinose, which is here, ARA, and PTET is activated by anhydrotetracycline, or ATC, which is right here. And you can see these are the individual transfer functions for PBAD and PTET. And these were measured using flow cytometry. So now the authors were able to produce an OR gate by placing both PBAD and PTET promoters in tandem, and they were able to see that when, as you can see from this image here, if either, here, let us just quickly draw the truth table for an OR gate. So this is OR. So if the first input, both are 0, the output is 0. If one input is on, the output is on. If both inputs are on, the output is also on. So this is the truth table for an OR gate. So if you can make sense of this, as the concentration of arabinose and anhydrotetracycline increases, you have a really strong fluorescent signal, and that shows that the OR gate is on. And as you move towards lower concentrations right here, you see that the gate is off. So now, to convert the OR gate into a NOR gate, the CI repressor gene is placed under the control of PBAD and PTET, and YFP is expressed from a second plasmid under the control of this CI repressible promoter. And then, as you can see from this image right here again, going back to the truth table, only at lower concentrations of ATC and arabinose, basically when both those inputs are off, do we get a strong fluorescence signal right here. And in any other case where you have either of the inputs on or both of them being on, we get low fluorescence signaling. And so this is the basic construction of a NOR gate. So this is figure 2 in the article, and figure 2 shows the characterization data for 3 different tandem promoters, PBAD, PTET, and PLAS. So they made combinations of PBAD, PTET, as seen here, and PBAD and PLAS, as seen here, and PTET and PLAS. So these OR and NOR gates, as shown before, use promoters as inputs, right? And so this feature allows these gates to be engineered in such a way so that they can respond to different inputs by replacing these promoters. So to investigate this, the authors swapped the input promoters of the logic gates, so they tried different combinations of promoters, and 3 are shown right here, and the output, the predicted output as well as the actual output is depicted in this figure. Now the promoter PLAS over here is activated by the quorum signal 3OC12 HSL. This is 3-oxo N3-oxo-dodecanoyl homoserine lactone. Now these gates perform as the additive combination of the individual transfer functions of these two input promoters and the CI repressor NOT gate. So the predicted transfer function for the 6 gates are shown right here. And now I think we already discussed the PBAD and PTET from the previous figure, but let's look at the PBAD and PLAS logic gate, and you can see you get similar results. Let's draw the truth table for the XOR again. So only when both of them are off do we have a signal. Sorry. So as you can see here, as at lower concentrations of both 3-OC12 HSL and arabinose, we get a really strong fluorescent signal, and at higher concentrations we have no signal at all. And if you move to PTET and PLAS, it's a similar situation as well. So this is figure 3 from the article, and basically what the authors are trying to say here are that complex logic gates can be designed using layers of simple gates. And so in this particular figure, they show the construction of, they show the construction of an XOR gate right here, built with 3 NOR gates and a buffer gate. And the output of an XOR gate is on only when either of the inputs are on. So you can see right here and right here. When either of these inputs are on, the XOR gate is on. But if both are off, it's off, and if both are on, it is also off. So what the authors did was that they took 4 strains of E. coli colonies, each carrying a different logic gate with different input promoters, and they were able to construct this XOR circuit. So as you can see over here, these strains are spotted onto an agar plate in the spatial arrangement, in a specific spatial arrangement required to perform the XOR function. And figure A over here is just an expanded version of the genetic circuit within each of these cells. So cell 1 carries a NOR gate that uses arabinose from the PBAD and N-hydrotetracycline for the PTET. And these are the inputs. Arabinose and ATC are inputs for cell 1. And it expresses last one as the output. So this cell is then wired to cell 2 and cell 3. The output of cell 1 is wired to cell 2 and cell 3 and acts as the inputs for cell 2 and cell 3 in addition to arabinose for cell 2 and ATC for cell 3. So I just said cells 2 and 3 use arabinose and ATC as their second inputs, and now their output over here is RH2, which produces C4HSL, which is N-butyryl homoserine lactone. N-butyryl homoserine lactone, HSL. So, and finally, cell 4 acts as the buffer gate and integrates the output from cell 2 as well as cell 3 by responding to the C4HSL. and the output of the buffer gate is only on when the input is on. So the complete circuit consisting of these 4 cells behave as a digital XOR gate with respect to the 2 inputs, arabinose and ATC. Now, each intermediate colony performs its digital logical operations appropriately, and it can be seen in and is seen with the output gene YFP. If you look at this figure right here, let's just look at cell 1. So when neither arabinose or ATC is present, since this is an XOR gate, you will get a high fluorescent signal, or that means that this is on. Now, when either of them are present, you get a low fluorescent signal showing that this is a normal XOR gate. And so the authors went and did that for the different cells, and finally when they looked at cell 4, it basically worked as an XOR gate. So when there was arabinose or when there was ATC, you got high fluorescent signal, or the XOR gate or cell 4 was on because you had high fluorescence from the YFP. So this is figure 4 and the final figure in the article, and the authors were able to construct a library of 16 simple logic gates, mostly using a combination of OR gates and NOR gates, as you can see over here. The circuit diagrams are for each of these gates are shown over here, and each circuit diagram is reproduced by the spatial arrangements of these different E. coli strains, as we saw for the construction of the XOR gate. Now, the authors were interestingly they didn't have to genetically manipulate any of these cells to get these gates. It was basically a simple combination of different gates, and they were able to produce a complex gate that would normally not be found that would not be found within a colony. And so also they noticed that there was no degradation in the signal due to the different layers of gates that were used to produce the output. So just in summary, the authors were able, through their experiments, demonstrated that a library of simple gates can be used to perform more complex computational operations by linking these gates using diffusible signals, such as arabinose, ATC, 3OC12HSL, as well as C4HSL. And the motif of multiple promoters in tandem, which was driving the expression of a repressor, is common, and so these NOR gates may represent a fundamental unit of biological computation. Finally, the authors point out that their current ability to create these logic gates within a single cell is limited. They may ultimately be able to encode more complex circuits in individual cells that are linked by cell-cell communication. And they suggest that these are the simple principles in the engineering of biological systems to create increasingly complex functions.